attendees are joining. Welcome to the Worldwide Neuro Seminar Series on Addiction. This is our second to last seminar of this season. Next week, we'll have Professor Véronique Desroches-Gamonet, who will be our last speaker for the summer. Then we'll have a break and we'll start up again in the fall with a brand new slate. So please look out for more seminars in September. But today we're very lucky to have Professor Peter Kalivas, and Céline Nicolas will introduce Dr. Kalivas.
Thank you, Marina. So it's a great pleasure to introduce Peter Kalivas today. So Peter received his PhD in pharmacology from the University of Washington and following postdoctoral training at the University of North Carolina. Then he became a professor at Washington State University before his founding the Department of Neuroscience at the Medical University of South Carolina in Charleston, where he remains a distinguished university professor of neuroscience. Peter is a neuroscientist best known for his work to elucidate the brain molecules and circuits that underlie addiction. His work is highlighted in more than 400 papers, and as editor of 6 books that he focused on the cellular mechanism and brain circuitry mediating psychiatric disorder. He received many awards, including Merit Award from National Institute on Drug Abuse, the Governor's Award for Research Excellence, South Carolina, and he is past president of the American College of Neuropsychopharmacology. And I cannot wait more now to hear about his talk because his title was very intriguing. So Peter, I give you the mic.
All right, I'm on. Yeah, thanks, Céline, for that very kind introduction. And I really want to thank the organizers. I mean, there's huge service to the addiction community to get this webinar thing going during a pandemic. I mean, so it's real kudos to you. You'll go down in history as saving addiction research. Okay, so I'm going to talk today about D1 and D2, medium spiny neurons in the nucleus accumbens. And I'll introduce to some of you are already very familiar with it, the concept of tetrapartite synaptic plasticity. And as far as we can, I will try to lay that on top of different functions for the D1 and D2 nucleus accumbens neurons. So, there it goes. All right, so I'm probably the
You're going to need to share your screen.
Most senior citizen in this seminar series.
You need to share your desktop. Share your presentation. It's not showing.
Oh, share it again. Okay, you took it off. I apologize. No wonder nobody knew what I was talking about.
You can't tell unless somebody tells you so.
I didn't realize it went off. Okay.
There you go.
Let's go back then. Uh-oh, I don't know if I can go back. Well, there was a slide that had this title on it, beautiful picture of South Carolina, and now we're on the second slide. Everybody can see everything okay? You're good? All right.
Yes.
So as I was starting to say, as near as I could tell from the speaker list, I'm probably the senior citizen of the speakers, or pretty close to it. So forgive me, I'm going to go back to the future to start the talk about some maybe new concepts, or at least the way these 3 concepts are put together might be new for most of you in the audience. And I think they're important. They've started to frame up our research, really. We didn't really even realize in many cases that it was being framed by these earlier concepts. But as we went along, we started realizing that these 3 terms really describe a lot of what we're doing. So the first is from this earlier publication by the famous duo George Koob and Michel Lemoine, and it's the term allostasis. So this is a more anthropomorphic term, mostly a behavioral term, as it was used in this particular review, to describe the difference between a homeostatic situation, which we would call normal. People adapt when they get a stimulus, they respond in an adaptive way. Allostasis would be an altered state where you get the same stimulus and you might respond in a maladaptive way. So allostasis, I mean, George and Michelle focused on addiction, so it's very good for addiction. But allostasis is a term that you could apply to most psychiatric disorders, if not all of them, that you're in an altered state, so that your responses to certain stimuli are not going to be adaptive. Before that, Abraham and Mark Baer published this TINS paper where they described or coined the term metaplasticity. And metaplasticity, the way we think about it in our group, is that it's kind of the same thing as allostasis. So I'll describe metaplasticity in a more functional way in a second. And then finally, and we'll deal with the first two first, and then about a third of the way through the talk, we'll start getting into the tetrapartite synapse. And this is something that came out of Dmitry Rusakov's lab, the term, and he's really the founder of this whole idea that we should be looking beyond just the pre and post synapse into other compartments. And so we'll go there, like I said, in about 15 minutes or so. All right, so this is just a metaphorical way to think about allostasis and homeostasis. So this is not, it looks a lot like a resting state functional connectivity of the brain, but in fact, this is, as is obvious, is a map of the world, and it's flight patterns. So you have your international and domestic flights that occur during the course of a 24-hour period. This is January 2020. So we would call this homeostasis. It's a stable state. People are flying back and forth, doing their work, having vacations, seeing families, etc. It's a very stable situation that works and is adaptive. Then by April, this was the situation. So this is an allostatic situation. We know, of course, that this is the result of an obvious disease state, the pandemic, but basically you now have stable communication between the other areas, but it's not as functional. And so this is an allostatic state that we would think of loosely as a psychiatric disorder, or in the case of what we're going to talk about today, addiction or substance use disorder. And it's stable. As we all know, it's been like this for a year. So it's a very stable state that individuals, or in this case, the entire planet, have to function in for a while. All right, so let's go with a more methodological definition. This came from a Nature Review Neuroscience paper in 2008 of metaplasticity. So you start in a homeostatic state, as I described, and then you use drugs, and you wind up in what I'm going to call allostatic metaplasticity. So it's an altered state, and there are changes in the brain, plastic changes that are associated with that. And they are enduring for the most part. They last a long time after you discontinue taking drugs. And this is what we would call substance use disorder. Then a lot of what we do is we expose the animal, and even in our clinical studies, we expose the people to drug cues, and we measure brain changes. And this would be called synaptic plasticity. So if you're in an allostatic state, you're going to get a different synaptic plasticity than you might from for example, a natural reward. And indeed, as we go through the talk, I'll mostly reference it. I don't have time to go through all the sucrose data, but if we substitute sucrose for drug use there, we don't measure significant metaplastic allostatic state. And certainly when they get their drug cue, they respond just like a drug-seeking rat operantly, but there is no synaptic plasticity that we could measure either in responding to a sucrose cue. So basically sucrose, you stay in the homeostatic state. Drug puts you into an allostatic metaplastic state that you then get an altered synaptic plasticity off of when you see a cue that drives a behavior forward. I forgot to mention Khaled. So this is the guy who got it going in the lab. He's sort of first realized that we were dealing with metaplasticity. He's now at the University of Pittsburgh. Okay, so this is the animal model, and the data I'll show you come from different variants of this model. Some of them exactly this model, others slightly different, but this is the basic model that we generate all the data I'm going to show you. And this is mice. We could do this with mostly heroin and cocaine, but we do it a little bit with cannabis, a little bit with nicotine. They're trained to self-administer, standard operant paradigm. Then we put them in extinction behavior or not. The bottom line is they get withdrawn from the drug for at least 10 days. And then we've associated a light and tone cue with drug infusion. And we present that cue in the extinguished state, and the animals pop up and start pressing the lever or nose poking for no reward. So it's not a rewarded cue responding. Nonetheless, as we all know, they respond very nicely on the operand and gradually extinguish that response if you leave them in there long enough when they realize the cue is no longer reinforcing or providing the reinforcer. So we have 5 groups in our most full-blown studies. So there's 3 that would be called drug groups and 2 that we would call sucrose groups. So the drug groups include a yolk saline, then there's a withdrawn animal, and they're either put in extinction or not, depending on the study. And then we have a cued seeking session where the cue comes back to the operand, but not the reward. And then the animals, we look at them 15 minutes later or sometimes 120 minutes later. As a control group, we have sucrose animals that essentially go through the same thing. And when we look for the plastic changes, the big ones that we find, we always run the sucrose groups to compare. And I can just tell you right now, all the stuff I'm going to show you does not happen with sucrose. So this is a drug metaplasticity dependent event, all of the cue-induced responding that I'm going to show. So if we want to look at long-lasting allostatic metaplasticity, we'd kill the animals right here. If we actually look at it in the extinguished environment, then we have some plasticity on top of that. But typically we look at it 24 hours after the last extinction trial or with no extinction training. If we want to look at cue-induced plasticity, of course, we'd look after exposing the animal to a cue. All right, so you missed the title slide, but the title had D1 and D2 MSNs in it. So let's start there with a little history from the lab on D1 and D2. So as you're all aware, D1s project in what's called the direct pathway, D2 project in the indirect pathway. Jin Yun, when she was a graduate student, she's now chair of neuroscience at the Medical College of Georgia, used in situ hybridization to show that this was happening, that there was a very significant D1 contribution to the indirect pathway from the nucleus accumbens, not from the striatum, the dorsal striatum. And that, nobody paid attention to that until Yoni published using some electrophysiology in transgenic animals many years later to demonstrate that indeed there was a highly functional D1 innervation through the indirect pathway. So we now know that both of those are important, and this basic anatomy was followed up on by Daniela, who's currently an assistant professor in the lab, and Jasper, who is now an assistant professor at the University of Colorado. And by the way, all the pictures you're going to see, people look a lot younger in some cases than they are now. So if you don't recognize them, that's why. They're typically pictures that we took while they were in the lab. Anyway, there were these 2 papers, and what they showed using DREADS and electrophysiology is that it's the indirect pathway that is most important for the drug-seeking event. And that if you activate the D1s, you promote drug seeking. If you activate D2s, you reduce drug seeking. And so that became, it was clear D1s and D2s can both modulate it if you activate or inhibit them. And we won't talk about it anymore, but it's the projection to the ventral pallidum that is most critical, the indirect pathway. All right, so more recently, Anna Clara, who's now at the University of Wyoming, wanted to look at ensembles and D1 and D2 ensembles in these mice that were responding for drugs or sucrose. And I'm just going to focus on the D1, D2 data. If you're curious, it's just an awesome paper, this molecular psychiatry paper she just had out, that describes her see her experiment. So these are the classic FOS trap animals, which means that when you expose the animal to a stimulus, it's going to activate neurons. It'll initiate CFOS, and at the same time, you'll deposit a reporter, an enduring reporter, in this case, tomato, in the cell. So you can come back later and weeks later if you want, and look and find the red reporter and see which cells were activated under, for example, a drug-seeking condition or in an extinguished environment. So that's what she did. And she went back and forth between sucrose. It's a really elegant study that it's worth looking at. I'm not going to talk about that part. I'm just going to talk about the D1s and D2s. So if you look at the cells that became tomato positive during a seeking event, this would be Q-induced seeking, or when they're placed back in the extinguished environment, what you can see is that in the seeking event, the Q seeking, the cells that show the tomato, i.e. were activated, CFOS expression was activated. So as a marker of cellular activation, you can see that it's mostly D1s. If you put them into the extinguished environment, so now they're bringing forward all the memories that they're extinguished in this environment, it's D2s are the predominant. Of course, you see some, the brain doesn't function like it does on DREADS. It functions where all the cells get involved a little bit. There may be predominant cell types, but even in seeking, you see D2s, and even in extinction, you see activation of D1s. So another way to look more directly at activation is something that everybody's familiar with by now, is the calcium imaging through these little mini cameras and a Grin lens. Then we've got the lens popped down into the nucleus accumbens, and we can look at calcium transients in individual cells. So that's essentially this experiment with a few traces. The data I'll show you has been generated by Redichal Hu. He's a MD, PhD in the lab, and he has a very complicated paradigm. We're trying to answer a lot of questions once we get the cameras working in these animals. But the heart of it for the talk today is in this protocol where you can see they get cocaine, and then we have abstinent period, then what we call the post-abstinent seeking. So they just get thrown back in the box. Some people might call it the first day of extinction. It's essentially the same thing. And then they get extinguished. In about 10 days of extinction, they're fully extinguished. And I'll show you calcium tracing or data from that second bar here. I guess you could see my pointer. Okay, so that's the first set of data, what we call post-abstinence. So it's a seeking event. And then there's the extinction event after they've been extinguished for a while. All right, so here's if we put the reporter, the calcium reporter in D1s versus D2 Cree mice. You can see the results. So the first thing you'll notice is that during a seeking event, there's a lot more cumulative transients. So this is just accumulated over the 120-minute set, a 110-minute session. How many transients occurred in D1 cells or D2 cells. So in post-abstinence, there's a lot of D1 activity. Once the animals are extinguished, it comes down. So it's in the extinction situation, there's much less activity. D2s stay about the same in both. So this is clearly, the D1s by this measure are clearly being affected by the extinction training, and they're showing less activity in the extinguished environment than they did in a seeking situation. Another way to look at this is you can look at the cumulative activity of individual cells. So this is, we take all the cells from this post-abstinence seeking and extinction time, and we put them all together with the time courses of activity. And then we do a little clustering analysis to statistically break apart the cells into different characteristics. And so in this case, we have three characteristics. We have those that are activated early in the session, and that's blue. So you can see a lot of these cells just jump up and show all their activity right away, then they kind of level off. You've got those that are activated later in the session, and then you've got those that were relatively continuous, which are shown here in the gold. So if you look at those three, it kind of goes along with what you would expect from a seeking event versus an extinction event. First thing you'll notice is that the red, which are the ones that come on late, is always larger in the D2s, whether it's a seeking event or whether it's an extinction event. So we assume that, especially here in the extinguished environment, that these are mostly D2s in this red category, And they're coming on during the within trial extinction. So here there's profound within trial extinction, but even in an extinguished environment, the animals nose poke a few times before they give it up. And so the idea here is that this activity is associated with suppressing the nose poking, and that's why it's gradually picks up as the session goes on. In contrast, you've got this early activity, which we assume is more associated with the drug-seeking event. And you can see that, especially in the extinguished environment, there's a little bit more in the D1s. In the seeking, both cell types show activity, which is kind of interesting. So it's not a clean picture. It's not what you can get with dreads or with optogenetics that, you know, everybody looks at and sees. When you actually look at the brain functioning in these situations, what comes out with dreads is a binary situation. D2s do this, D1s do that. There's a lot of different kinds of activity. And so to deconvolute that a little further, you're probably familiar with this. You basically time lock the cells around the nose poke. And then you look at cells that are activated or inhibited around the nose poke. So this would be examples of a bunch of cells that are activated around a nose poke. These are a bunch of cells that are inactivated around a nose poke, up to a threshold, a Z-score threshold that we consider significant. Bottom line is we have something very similar. You got some cells that are excited, some cells that are inhibited, some cells that actually do a little bit of both, depending on which nose poke it is. Okay, that's the percentage of cells that are showing some time lock responding, D1s, and you can see there's less of them in extinction. In D2s, it goes down a little bit, but not as much and not significantly between the seeking and the extinguished environment. So in this case, again, it's not as black and white as the DREAD studies are, but it looks like the D1s are being less active in the extinguished environment, whereas the D2s tend to show activity in both, even in response to a nose poke. And the caveat, of course, is that you're getting about 40 nose pokes here, and you're only getting 8 nose pokes here. All right. So we would conclude in broad brushstrokes, the D1 MSN activity is associated with drug seeking, D2 MSN activity is associated with extinguished behavior. All right. So now how does all that fit in with that other term that Dmitry Rusakov got started, which is the tetrapartite synapse? So, you know, I'm not going to get down on normal synaptic plasticity. It's done wonders for the field of neurobiology over the last 70 years. I mean, all sorts of discoveries have been made. We know an awful lot now about how the brain works. And pharmaceuticals have come out of understanding what synaptic activity is and the neurotransmitters that are involved, et cetera. So it's been hugely valuable. However, if you just take the neural peel at large and you look at it, it's mostly black matter if all you do is highlight the presynapse and the postsynapse. So just like the universe, we don't really know what the dark matter is, but in this case, we do. And so in order to really see a complete picture, the idea is that we have to look at 2 other compartments that surround the pre and post synapse that are equally, and as I'll try to indicate, in some cases more involved in the allostasis or metaplastic state of drug addiction than the pre and post synapse are. So the other 2 compartments are the Astroglia, which come in close to the synapse, and express proteins right near the synapse. We'll talk about that. And the 4th compartment is the extracellular matrix, which is a proteinaceous structural, but also signaling domain, as we'll talk about, that go around all the cells, including inside the synapse. All right, this is what it really looks like, of course. You can see the presynapse with the synaptic vesicles, the postsynapse with the density there opposite the vesicles. And we color-coded on the right. Anna did all this coloring with the different compartments in different colors. So I'm going to kind of do this backwards. We started with the postsynapse like everybody else, but I'm going to introduce that late because actually that's how the brain gets to it. when it adapts to addictive drugs. Okay, so let's start talking about Astroglia first. So GLT1 is a critical protein that Astroglia express. Maybe there's a little bit in neurons, but it's almost 100% in Astroglia. And it's expressed in a patterned way such that most of it is right at the synaptic cleft. And of course, the reason it's there is to rapidly eliminate synaptically released glutamate. So to promote the fidelity of synaptic communication, you don't have glutamate spilling out all over the place. So one of the fundamental allostatic metaplastic adaptations that we stumbled across, and it's been replicated many times in many laboratories, and it happens with most of the addictive drugs that have been looked at, is a downregulation of GLT1 in the nucleus accumbens. This is old data. It's kind of a summary slide with using Western blots. And in the old days, we also did uptake assays, so to get at a functional measure, and that's downregulated in the nucleus accumbens. This work was looking at glial transporters, was started by Dave Baker, who's now at Marquette, and then Lori and Kate really brought in the GLT1 big time. Lori's now at the University of Florida, and Kate's at the University of North Carolina. And importantly, it's downregulated metaplastically, because it's enduring, but it can be reversed by a number of treatments. So you can give N-acetylcysteine, ceftriaxone, propentafine, there's probably others. And that restores the levels of GLT1 and prevents cue-induced reinstatement of nicotine seeking, heroin seeking, cocaine seeking, etc., cannabis seeking. So we know that it's important, and it's a critical metaplastic adaptation. Well, as I mentioned, GLT1 is expressed almost entirely by astroglia at the synapse. So we realized we needed to take a much closer look at astroglia, look at astroglial morphology in particular. So Mike was in the lab at the time. He's now an assistant professor here at MEOC in anesthesiology, and he developed an approach that allowed us to inject, basically transfect a membrane-targeted reporter. And then we could digitize this, isolate, mask out other cells, digitize the cells, and really take a good look at the morphology of the astroglia using something as simple as confocal microscopy, not serial section EM. And you can see that the morphology, if you look in panel C there, is ridiculous. So it makes studying synaptic spines super easy. This is a very complicated morphology. It looks more like a sponge than it does like a structure, a solid structure. Then Anna picked up on this, and what I'll be showing you is her data, both what's in that biological psychiatry paper there and on into stuff that she hasn't published yet. Okay, so this illustrates some of the stuff that we do with these digitized neurons, or glia, and this is just a little movie. You can see the vessel kind of going through in that one spot right there. And you can also see the very complicated morphology. What you'll notice here is there's purple dots on it. And those purple dots are essentially a counter stain. And the data I'll show you are two of the counter stains that we use. One is synapsin-1. So we use it as a biomarker for the synapse. It's expressed only in the presynapse. And the other, of course, is GLT1, as I've already talked about. And in this way, we can look at how much is associated, how much the glia gets within 250 nanometers of synapses. And in this digitized version, that's a purple dot there. We can also look at surface expression, for example, of GLT1 and whether it's associated with synapsin as well. So in other words, it's close to the synapse. So lots of things you can do once you get this thing working. It's not trivial. And so the first thing is we replicate the basic metaplastic finding that I showed you earlier. So these are our 4 treatment groups, and it's been done with sucrose. It doesn't happen with sucrose. And you can see that GLT1, the total GLT1 in a glia, these are all individual glia, these circles, is downregulated after extinction, and it's metaplastic, so it kind of stays downregulated the whole time. It's not something that's going to necessarily change. But if we looked at surface expression only, and that was a little tricky, but Anna got it worked out. So this just shows, without going into detail, how we can isolate the surface and then do the labeling needed to look only at the surface. What you see is that the surface expression, even though total GLT1 is downregulated in the withdrawn extinguished animal, surface expression is maintained. So they're doing everything they can. It's just not in the synapse anymore. It's kind of out there on the fringe, which I don't show those data. The thing is that when they see a cue, there's a plastic event that takes place. It's transient, because you see we kill the animals while they're actively responding for the cue at 15 minutes, or after they're pretty much done responding for the cue at 120 minutes. That increase in surface GLT1 actually goes back to the earlier state. So this is a plastic event that is produced during by the cues. And this is that's actually unpublished data. This is what was in that biological psychiatry paper. You can see that you get something very similar with looking at synaptic proximity of the astroglia, using that approach where we labeled with synapsin 1. So you can see first is that there's a decrease in synaptic proximity after heroin. This is all of Anna's work is with heroin. Self-administering rats. Oops. And what you'll see is that it doesn't look like much, but basically it's no longer significantly different from the control animal, because a portion of the astroglia have increased their synaptic contact. So they've kind of gone back to the synapse. And then as with all of this kind of cue-induced plasticity, it comes back to normal after 120 minutes. So first thing is we've developed ways, I don't have time to go into it, that if we prevent the astroglia going back by knocking down a critical glial-specific actin binding protein called esrin, animals reinstate more. So they have a greater cue reactivity. Likewise, if we prevent the GLT1 from going back by a more standard knockdown of just GLT1, we accentuate the reinstatement event. So these things that are happening during cue by the glia during the cue are compensatory. But as I'll make the case, the metaplastic changes are bad. They're the ones that cause the reinstatement event to occur. All right, the first thing, it's just kind of an aside, it's very cool, and it led us into stuff we're doing now that we haven't finished, but we're pretty pumped about it, so I'm gonna show it. It looks like we take all the glia and put them together from sucrose animals, et cetera. The reinstating animals, 15 minutes of reinstatement, cued reinstatement, are the ones that are showing the increase in surface proximal glia, GLT1, or synapsin co-registration with the astroglia. So these are all reinstating animals. The sucrose guys are all stuck down in here. And there are two populations. So if you just do a clustering analysis to identify subclusters, you can see that one shows the increase in GLT1, the other glia show the increase in synaptic proximity. So it's almost like there's two types of astroglia. So this caused us to really think about a study that Alfonso Arac did, published it in Science a couple of years ago, that said that in the striatum, astroglia tend to associate either with D1 or D2 MSNs. They don't typically associate with both. So that, obviously we got two groups here that are undergoing this cue-induced plasticity. Maybe that's with D1s versus D2 MSNs. So we're just getting this off the ground. But you can see we've pretty much got a good end in this particular treatment group, which is the cued heroin-seeking group. And we're looking both at the GLT1 on the surface and the synaptin co-registration with the glial membrane. And you can see it's much higher in both cases in the D2s than in the D1s. So the synaptic coverage and the return of GLT1 appears to be more on D2s than D1s. Now, we really won't know that, of course, till we do the withdrawn groups and the saline groups and all that. But there's definitely a difference between D1s and D2s just in this group. So that's very exciting. That's the future. All right, so GLT1 downregulation leads to spillover of glutamate. This was something we discovered early on. Krista really did the first study looking at spillover during a reinstatement event. Then Ryan went with Coke, Ryan did it with heroin, Cassie, who you heard last week, did it with nicotine. And other people have looked at this as well. So it's pretty established event. And so, when you have a drug cue, and all of these studies showed that the prefrontal cortex input was the critical glutamatergic input to driving forward this drug-seeking behavior. You could either do that optogenetically, or back in the old days, we just injected GABA agonists into the prefrontal cortex. Anyway, this will initiate a drug-seeking event. It'll initiate a sucrose-seeking event. In the drug state, there's been a metaplastic change in these synapses. GLT1 has been pulled away from the synaptic cleft by this retraction of glia, which is enduring. Glutamate spills out, and all these studies were dialysis studies. There's other folks have done some biosensor studies, and they show this massive spillover of glutamate during cued drug seeking in the accumbens, but not cued sucrose seeking. All right, so just establish that as a fact, and it's mediated by this withdrawal of glia from the synapse. So what does it do? I don't have time to go into this. It's a really elegant study that Alex did, who is coming back to MUSC from Mount Sinai. We're very excited about that. He'll get here sometime in the late fall. But what Alex showed in this paper is that spillover stimulates mGluR5 receptors on nNOS expressing interneurons. So this is neuronal nitric oxide synthase expressing interneurons. If you increase calcium through a GQ receptor like mGluR5, you're going to activate nNOS synthesis. In nNOS, and you'll activate nitric oxide synthesis. So now you've got nitric oxide. Just as you know, nitric oxide is very lipophilic. It goes everywhere like a gas would. And so it's definitely made its way into the extracellular matrix. So now you're filling up the extracellular matrix with nitric oxide. So what does that mean for signaling out there? And so now we're going to move on to the extracellular matrix. All right, the extracellular matrix, as I mentioned, is a signaling domain. So it's like the intracellular space. It signals. When stuff happens, including synaptic activity, it signals. And how it signals is catalytically. So basically, enzymes activate that then cleave proteins that make up the extracellular matrix. And once those proteins are cleaved, they then bind differently to cellular membranes, to different receptors. Key among those that we're going to talk about is, for example, integrin receptors. So MMP9 in particular has been studied early on in like LTP and things like that. So we know it was important in classic forms of synaptic plasticity. But how does that all get to nitric oxide? Well, they're metalloproteases, which means they have a zinc that the protein is built around, and the zinc can bind in one of four places. If it's bound in these three, the protein's inactive. If it's bound over here, if it's bound in one of these three, the protein's active. If it's bound over here, it's inactive. It tends to spend its time more in the inactive state than it does in the active. So this is the favored configuration with the zinc bound right here. But it flutters just like all isomers and things do. So if nitric oxide is around, it will nitrosylate that site, preventing the zinc from coming back and forcing the zinc to remain in the active configuration, which activates the enzyme. And Alex in that paper showed that for sure this is happening with MMP2, MMP9. Our reagents weren't good enough to really get a strong signal off of the nitrosylated protein. Okay, so this is how we think that little cycle of NO makes it back to activating the matrix. What we do know for sure is the matrix gets very activated, starts signaling like crazy through this MMP2 and MMP9. And so how Alex showed that is that you basically put a false substrate into the nucleus accumbens, these are gelatinases, so it's basically gelatin, and it's what we would call Fitzy quenched gelatin. So it's got a fluorescent Fitzy that is quenched inside of it. And as it starts getting digested by the activated MMPs, it begins to fluoresce. And it does so in a linear fashion over time. So the more fluorescence you see, the more MMP activity there is. So the micrographs are obvious. If you just look at the bar graphs, what you'll see is that there's some activity in which we would call metaplastic activity in the extinguished animal. And there's also plastic activity that's even more dramatic in the reinstatement animal. And like all the other things I've shown you, it's transient. So as the animal quits reinstating, responding to the cue because they've extinguished it and within trial, the MMP activity goes down. And you can see something similar for nicotine and heroin. Of note, at least with this measure, there was no metaplastic change with the heroin, but there still was the synaptic plasticity that takes place after. Okay, so it gets activated. And Alex showed that this was MMP9 here, and this is MMP2 here. So Vivian, who's an MD PhD and is now finishing up medical school, did this awesome study just recently. And she basically refined that protocol, the in vivo zymography, which allows you to measure MMP activity, using some of the masking procedures with the confocal microscopy that we use for glia, and was able to isolate the activity, the MMP activity, within 250 nanometers of dendritic spines and dendritic shafts. And then you just, of course, use Cree animals, and you can look at the D1s and the D2s independently. And so this is the primary finding from that paper. So let's take a look at the D1s and the D2s. So you can see a yoked, extinguished, and reinstated. And I should mention that it comes back down. I don't show that here, but if you kill at 120, everything's back to normal. The D1s show nothing in terms of MMP activity after drug use until they see a cue. And then you get this big increase in MMP activity, and it's MMP9, as she went on to show, around the D1s. Very different around the D2s. The D2s show a constitutive activity, so an enduring activity after they've been withdrawn and extinguished. That's MMP2s. And interestingly, when they see a cue, that shuts off. And she went on to identify with the TIMPs that are involved in shutting it off. So you have a metaplastic event, which is this D2 MMP activity that's constitutive, existing days after withdrawal. But you also have a plastic event that occurs in both cell types. The D1s go up and the D2s shut off. So again, both of these are going to be involved in the reinstatement event, but this is a more metaplastic change. Okay, so now let's do the postsynapse real quick. Hopefully I'm not running out of time. I'm trying to check now. Looks like I got a few minutes. Okay, so this is older work, as these dates would indicate. And these are the three main people. A lot of people helped, but these are the three main people. Started off with Hao-Wei Shen, who's now at Ningbo University. Yoni picked up the electrophysiological arm of it, and he's at Hebrew University now. And Cassie, who you heard last week, sorry, Cassie, if you're there, I missed your talk, so I don't know if you talked about this or not, but is at the University of Kentucky. So we know now that this is almost entirely occurring on D1 spines, not D2, this postsynaptic plasticity. There is some things that happen on the D1s, but the overall, or D2s, but the overall event looks like it's D1 spines that are contributing to these measurements. Okay, so here's what happens when the animal sees a drug cue. So the spine heads expand, and there's AMPA receptors measured by AMPA and MDA ratios inserted postsynaptically. In other words, you got bigger, stronger spines. And so in theory, synaptic communication has now been amplified. Then, as the animal is within trial extinction, in other words, you kill them at 45 or 120 minutes after starting the reinstatement session, the spines have shrunk back and the ampin and MDA ratio goes down. So you have this expansion, this potentiation basically, that then poops out. So we called this transient synaptic potentiation at the time. And as I'll describe, this is built on that metaplastic event in the Astroglia. Okay, what if you do a sucrose cue? And we ran through all of these things with sucrose. This is what happens, nothing. We can't measure anything happening in the sucrose trained animals. So it's tied in with that whole cycle of things that I've talked about. All right, just a lot of work was just recently done by Coty to prove, to link this event to MMPs. And Armino is the first one way back when she was an MD PhD student, she's now at Penn, was that identified beta-3 integrins as probably important, but it was very indirect. And Coty's gone on to show that it is definitely beta-3 integrins is critical. It interacts with the matrix, and that's why it's important. And unearthed, so there it is interacting with the binding domain, the gelatin binding domain that the MMP9 revealed. And she worked out the full signaling, or almost all the signaling pathway for the spine head expansion. And we didn't get as far with, we know that integrins aren't binding as necessary for the ampin and MD ratio to go up, but we did not work out the signaling. It's definitely not PFAC. So that's contained in those two papers, where she also showed that the spine head expansion, as we know, and this signaling pathway are effective in D1s. So that's where you see this thing happening. You do not see it happening in D2s. Okay, so now we have the link all the way around the circle. So there's this summary slide. If you give a sucrose cue, glutamate transmission presumably is activated as the cortex responds to the cue, recognizes it as important, goes down, engages sucrose seeking through the nucleus accumbens. Because GLT1 and glia are normal, training animals on sucrose, at least the way we do it, does not produce any metaplasticity, so it's normal. The glutamate gets taken up, doesn't spill out, and the animals go ahead and seek sucrose. What's happening with the drug cue, and for sure this is heroin and cocaine, virtually every step has been shown with those two drugs. The drug cue spills, the glutamate spills out, so you still have that same basic awareness of going to get the drug, but now you have this amplification that takes place. And as I showed you, it involves spillover, or as I described to you, I didn't actually show you the data, it involves spillover to nNOS interneurons that then increase nitric oxide synthase. This is at least a prime contributor to the MMP activation that's occurring. This catalytically cleaves gelatin motifs, creating this ligand that's called RGD. And that leads to the synaptic potentiation and the amplification of drug seeking. All right, so what this has done is, at least as far as cue reactivity, and looks like context as well, activating animals or people, presumably, with drug-associated stimuli causes this to happen. And in our rodents, we can inhibit this by either restoring GLT1, which I referred to. I didn't show you the data, but any of these things in all those papers contains these experiments. Any of these angles, specifically in the nucleus accumbens, will prevent drug seeking. And we moved the GLT1 restoration into clinical trials, and it inhibits the cued craving, not so effective on reinstatement. Some yeses, some nos on that. I mean, on actual relapse to drug use. And I don't have time to talk about what those problems might be. There's a whole host of other sites, and maybe combinations, et cetera, will be more effective. Well, as you've gathered from all the pictures I've shown over over the course of this talk, I'm about the luckiest person on the planet. You know, I've just had a fantastic time over the last 35, possibly even 40 years, I'm not saying, that working with just really smart, creative, hardworking people. This is the most recent crew. We've also had the benefit for the last 15 years or so of having a NIDA center here. And these are the people I see. I did not update that slide. So there are a few other people in that center and a few people that have moved on. So I apologize to Hei-Shing if he's looking at this, and Catherine. Their names should be up there. But bottom line is we would not have gotten nearly as far in this convoluted thing that I just showed you if we hadn't had these people egging us on, encouraging us, et cetera, and really making some of the insights on our behalf that I described to you. And I've had the very good fortune of being funded. This work I'm showing you is primarily out of NIDA, but with some contributions from the VA and NSF. So I was a chair for 20 years, no longer, and loving it. But what I want to say about that is there's some good things. So those of you that have aspirations, I'm sure you're out there that really want to be chair someday. There's some great things if you're given a license to build and really bring in some brilliant people into your department. The rest of it is not so entertaining. At least I didn't find it to be so. But one of the things you could do is I could have a Halloween party, a departmental Halloween party. And this is, of course, post me being chair, because you can see we're at a local pub for our lab Halloween party because the department quit having a Halloween party. So if my chair is watching this, maybe this will convince him to reinstate the Halloween party. All right, thank you very much. It's really been a pleasure. I think there's time for questions.
Thank you so much,
So
Peter.
should I
that was a great
get
talk.
this off or some? Stop sharing?
Yeah, probably stop sharing just so people can watch you. And Celine will start with the questions in the Q&A. Anyone who wants to ask a question, please do post it in the Q&A box, and Celine will read it out. And we do have one in there now.
Yeah, thank you so much for your talk, Peter. So I'm going to read the first one from Xinju Wang. So thank you for the great talk. And it's a question about the calcium imaging data from the beginning of the talk, and asking if the baseline neuronal activity of D1 and D2 MSN altered after abstinence, or only their response to drug-associated cue is altered.
Yeah, that's a very good question. And one of the reasons we stated that abstinent period in that particular paradigm is because with the cameras on, we get some, but it's a blunted cue-induced reinstatement in our mice. So that's why we put in that abstinence period. What I've seen, and it's too bad Retta's not here, he might have a more detailed answer. We do a cue-induced reinstatement at the end, just by cue alone once the animals have been extinguished. And I believe we get the same pattern where the cue causes the increase in D1, calcium activity, just like this other seeking event does. In other words, it's on par with that. That's what I've seen. We haven't accumulated that data to the extent. So whether it's a combination of context plus cue or cue alone, I think it's the seeking event. It's the motivational state that is causing the D1s are contributing. I mean, it's a chicken and an egg, but that is associated with the increase in D1 activity.
Okay, thank you.
I had a related, sorry.
Go ahead, sorry.
I was just going to ask about the glutamate inputs to the D1 versus D2, and the fact that the synaptic changes onto the D1 are really the ones that are most clearly regulated over the course of drug exposure and then into relapse. Is it known why those synapses are particularly susceptible to plasticity and the D2 synapses are not? I'm always puzzled, why not the D2?
Right. And so the D2s are clearly involved. I mean, we show the changes in activity and everything, and that's so they're clearly involved, if nothing else, in shutting off the drug seeking. And if you place an animal into an extinguished environment, there is actually an increase in AMPA and MDA on the D2s. I didn't talk about that. So there's a little bit going on, but that whole full-blown syndrome does not occur. I think we're getting at the answer to that when that one slide that I said, we're all pumped up about with the D1, D2, and the glia.
Yeah.
It looks like the restoration or the compensatory event that the glia are doing, which is jamming the GLT1 back towards the synapse, and by the process going out towards the synapse during the cue, it's shutting down, or it's preventing the spillover around the D2 relative to the D1. And so we're thinking that might be the reason, but as you got, we're in the primordial days on that one, and that's, we're hoping for a science paper someday based on that.
Great. I have another question. Can neuroimmune factors affect the neuroplasticity in addiction? And additionally, other than GLT, do you think astrocytes use any other receptor or pathways to induce neuroplasticity?
I'm going to have to ask you to repeat that question. I apologize.
No, it's totally fine. So there is two actually.
Yeah, speak slowly, and then I'll get it for sure.
Can neuroimmune factors affect the neuroplasticity in addiction?
So one more time.
Like
I
neuroimmune factors like microglia.
Oh, okay. Neuroimmune. I'm sorry. You speak fine, by the way. It's my hearing. Yeah, I think so. But we haven't been studying those at all. I mean, there's lots of evidence that you can manipulate the immune system in the brain and do things to the extracellular matrix, for example, et cetera. So I think they're involved. We haven't worked them in. Kate Reisner, one of the things she did when she went off to UNC was to start working on that aspect of the project. But we have not been doing it.
Thank you. So the other part of the question is, other than GLT, do you think astrocytes use any other receptor or pathways to induce neuroplasticity?
Yeah, so that's an active area of research, and basically we assume that it's the spillover interacting with mGluR receptors on the astroglia that are causing the astroglial synaptic plasticity. Just like it increases mGluR nitric oxide synthesis on the interneuron, we are suspecting that that's the signaling pathway. It becomes difficult to work with, so it's not easy to work with. I mean, we just have to have the right reagents. So we developed glial targeted mGluR5 knockdown reagents and things like that, so that we can now start really looking at that. But we don't really have any data with those reagents. We tried just injecting antagonists and stuff, but they're not going to be cell specific to start with. And that acute jab into the brain mushes the glia around a little bit, so we don't get as clean a picture on what the glia are doing if we've just stirred up the brain with a microinjection.
Thank you. Another question is, how would you integrate dopamine terminals? Shouldn't we talk about Tetrapartite synapse?
The which terminal, sorry?
Dopamine? How do you integrate?
Yeah, so that's a good question. Again, as far as we're concerned, dopamine does not exist. But the reality is, of course, is that it does. And so we think of it mostly as follows, that the cue obviously increases dopamine transmission in the nucleus accumbens, but in our measurements or our studies, it seems like the release in the prefrontal cortex is more critical to the relapse event. These are older studies. We haven't gone back to it, but if we use something like fluphenazine, non-selective dopamine antagonist, you inject it into the prefrontal cortex, you prevent reinstatement. If you inject it into the core of the nucleus accumbens, not the shell, the shell does prevent reinstatement if you inject it into the shell. If you inject it into the core, it does not prevent reinstatement, at least in the simple model that we have. So we've always thought that dopamine is acting as kind of this initiator of the event to get the glutamate system going. So it's part of the brain's recognition system that a relevant reward or a reward associated stimulus has entered their world, and that involves getting the prefrontal cortex involved and the amygdala as well, of course. And those inputs to the nucleus accumbens core are what are driving forward the behavior. I think the shell is more associated with limbic structures, as you know, and that's probably part of the recognition event. That, the brain realizing that something important has just entered the world, and there's a squirt of dopamine that gets associated with that.
I'm afraid we're going to have to leave the last word on dopamine.
No!
No. Thank you so much, Peter. It was really a pleasure to hear your talk. Remember that the last Worldwide Neuro Addiction Seminar for the season is next Thursday at the same time. Thank you all for listening, and we look forward to seeing you again in the fall after next week.
Thank you very much. I really enjoyed it. Yeah, see you later.